Muscular physiology and the neuromuscular junction. Some universal characteristics of muscle would be excitability or responsiveness. They respond to chemical signals, stretch, and electrical changes across the plasma membrane. Conductivity, local electrical excitation sets off a wave of excitation that travels along the muscle fiber. Now, muscle fiber is what we call a muscle cell. So an individual cell is really elongated and will span the entire length of that muscle. When we talk about a local electrical excitation, that's very similar to when we talked about the nerve impulse. Contractility shortens when stimulated. Extensibility, capable of being stretched between contractions. And elasticity returns to its original rest length after being stretched. Skeletal muscle is voluntary striated muscle, usually attached to bones. So if you remember from general EMP, striations are talking about those stripes that you see in skeletal muscle tissue. Striations are alternating light and dark transverse bands that result from the arrangement of internal contractile proteins. They're voluntary, usually subject to conscious control. The other muscle types are involuntary. So if you remember what the other muscle types are, they're smooth muscle and cardiac muscle. So those two are involuntary while skeletal muscle is voluntary. Average size 100 micrometers to 3 centimeters, long but can be much thicker, and as long as 30 centimeters. Skeletal muscle cells, also called muscle fibers or myofibers. So this is a picture of skeletal muscle tissue. You can see the nuclei here, and these are those striations. So you can see the bands of light and dark. And those striations, I want to talk about what actually creates those and lay out all of the muscle fiber components. So the components of the muscle fiber, we have the sarcolemma. The sarcolemma is a plasma membrane of the muscle fiber. Here. So this is a picture of the sarcolemma. Sarcolemma is just the cell membrane. Then you have cytoplasm of a muscle fiber. Inside of the cytoplasm, you're going to have myofibrils, long protein cords that occupy much of the sarcoplasm. Glycogen, which is a carbohydrate stored to provide energy for exercise. And myoglobin, red pigment, provides some oxygen needed for muscle activity. So myoglobin can actually store oxygen within the muscle so that when energy is needed for a muscle contraction, we have that oxygen store ready to go. Many mitochondria bean-shaped near the sarcolemma and deeper ones are tubular and dynamic in shape. Multiple nuclei, averaging 30 to 80 per millimeter, serve the role in fiber repair. So going back to here you can see there's a bunch of mitochondria studded throughout, and then there's also a bunch of nuclei within one cell. So these are multinucleate cells, many nuclei. And why do we need to have multiple nuclei? Well, if you think about it, muscle fibers are made up of protein. And in order to make more protein, you need the instructions that come from the DNA. If you have a 30 centimeter long cell and there's only one nucleus on the end, how would you then be able to make the proteins and ship them all the way to the other cell? That would be really inefficient. So in order to increase the efficiency of creating new proteins, and these proteins inside here are constantly getting destroyed. Anytime that we contract, or if we are trying to do bodybuilding and we overextend the muscle and we rip the fibers inside of there, they need to be replaced pretty quickly. Otherwise, that can cause some pretty significant inflammation and prolonged damage and can decrease the muscle contractility. So we don't want that to happen. So we've got to be able to replace all of these proteins inside of here. And in order to do that, we have to have multiple nuclei, because if you remember the process of transcription and translation, transcription is when you make that messenger RNA copy of the strand of DNA that's in the nucleus. And then translation is when it goes to a ribosome and helps to produce that protein. So if we only have one nucleus, it's not really efficient. If we have multiple nuclei, we've got multiple little protein making stations or instruction stations so that we can make the proteins and repair those. So more components of the muscle fiber, also known as the muscle cell. The sarcoplasmic reticulum. The sarcoplasmic reticulum is a smooth endoplasmic reticulum that forms a network around each myofibril. The main goal of this sarcoplasmic reticulum is to store calcium for the muscle contraction. So a lot of people think, oh, calcium is needed in order to make strong bones, and that may be true, but calcium is needed for so many other things in the cell, in the body. So we learned about the nerve impulse and how in order to release neurotransmitters from the end of a neuron, a lot of times calcium has to enter. If we don't have calcium or calcium isn't available, we don't have nerve impulses. Now, is it more important for your body to have strong bones or to have nerve impulses? Obviously, nerve impulses are more important. But also, as we go through this lecture series, you're going to see that calcium is needed for every muscle contraction. And it's not just muscle contractions for skeletal muscle for us to be able to move. It's muscle contraction for cardiac muscle too, in order for you to have a heartbeat. So your body really does have to prioritize. When we are calcium deficient, we would actually break down our bone to release calcium into the body so that the calcium can then be used for these various super important tasks like having a heartbeat, fueling the muscle contraction or aiding in the muscle contraction for your respiratory muscles to move, your diaphragm, your internal and external intercostals. So calcium is needed for so many other things than just building strong bones in the body. The terminal cisterns, they're dilated ends of the sacs of sarcoplasmic reticulum, which cross the muscle fiber from one side to the other. They act as a calcium reservoir, increases calcium through the channels to activate contraction. Within cisterns, calcium is bound to a protein called calsequestrin. Cal for calcium, sequestrin. If you sequester something, that means you're absorbing it or sucking it up. So calsequestrin sequesters calcium or sucks it up and retains it. Transverse T-tubules. Tubular infoldings of the sarcolemma. Remember, sarcolemma is the cell membrane, which penetrate through the cell and emerge to the other side. Each T-tubule has terminal cisterns associated with it on either side. And those aren't very well pictured here, so we will talk about those once we get a little bit further on with the pictures. Myoblasts are stem cells that fuse together to form each muscle fiber early in development, each contributing one nucleus. Satellite cells are unspecialized myoblasts remaining between the muscle fiber and endomysium. They play a role in regulation of damaged skeletal muscle tissue. So when we look at the other components of this picture, so we already talked about the sarcolemma, which is just the membrane of this cell. Then we have the sarcoplasm, which is like the cytoplasm of this muscle fiber, and it contains all of the different substances inside. Then we have, here we see the triad, the terminal cisterns and transverse T-tubules. And those terminal cisterns are here, and the transverse T-tubules go straight through from one side to the other. Then we have sarcoplasmic reticulum. So the sarcoplasmic reticulum is here. And then we see the I band, the A band, the Z disc. The Z disc, we'll talk about that. That consists of actin or thin filaments. And then we have the I band, which is the lighter band, and the A bands, which are darker bands, and that's what helps make that striation pattern. Then we have myofibrils, which are packages of myofilaments, and myofilaments are the individual filaments that are going to be pulled inward past each other in order to shorten the muscle. So myofibrils are composed of muscle filaments or myofilaments. There's three kinds of myofilaments. Thick filaments, which are made of several hundred myosin molecules. And myosin is a motor protein. This is fueled by ATP and it allows for the contraction of the muscle. Each molecule is shaped like a golf club, two chains intertwined to form a shaft-like tail and a double globular head. Heads directed outward in a helical array around a bundle. Heads on one half of the thick filament angled to the left, while heads on the other half angled to the right. Then there's a bare zone with no heads in the middle. So this is what it looks like, and when they said it looks like a golf club, this is what they meant. Then you have thin filaments. Thin filaments are composed of three different protein types: fibrous actin, tropomyosin, and troponin. So fibrous actin, two intertwined strands of globular actin subunits, each with an active site that can bind to the head of the myosin molecule. Tropomyosin each blocks 6 or 7 active sites on G-actin subunits. And troponin, small calcium binding protein on each tropomyosin molecule. So this shows the molecular structure of the filaments. So here's the thin filament, and you can see here is tropomyosin, and then you have troponin, and then in the background these big red bubbles are going to be actin. So actin has this cross bridge of tropomyosin. Tropomyosin blocks the active sites under a normal resting condition. Troponin bind calcium. So troponin are here to bind calcium. When calcium binds to troponin, it actually causes these to move and unblock those binding sites. So right now they're covering all of the spots where myosin could bind to actin. But as soon as calcium binds, it kind of shifts this out of the way, exposing all of the myosin binding sites, and now myosin heads can actually bind to actin. So here you can see the bare zone in the middle. The bare zone doesn't have any overlapping actin. Here you can see the actin, troponin, tropomyosin complex on one side and on the other side. And the goal here is for the myosin heads over here to be pulling this actin toward the center. And the myosin heads over here are going to be pulling this actin toward the center. So they are going to be acting in opposite directions to shorten the muscle. If they were going in the same direction, then we wouldn't have appropriate shortening. So there's this bare zone in the middle, and the goal is for that one and that one to be pulled toward the center. There's elastic filaments, which are made of a huge springy protein called titin. They run through the core of the filament and anchor it to the Z-disc and M-line and help stabilize and position the thick filament. It prevents overstretching and provides recoil. Of the myofilaments, myosin and actin are the contractile proteins. They do the work of the contraction. Tropomyosin and troponin act as regulatory proteins. They act like a switch that determines when the fiber can and cannot contract. Contraction activated by the release of calcium into the sarcoplasm, and it's binding to troponin. Troponin changes shape and moves tropomyosin off the active sites on actin. Several other proteins associated with myofilaments to anchor, align, and regulate them. Dystrophin links actin in outermost myofilaments to membrane proteins that link the endomysium. Transfers forces of muscle contraction to connective tissue, ultimately leading to a tendon. Genetic defects in dystrophin produce disabling disease, muscular dystrophy. So this is a diagram. You can go ahead and pause the video and review all of these components. But here you can see the contractile unit of the muscle, The sarcolemma is the plasma membrane, and then there's some endomysium. This is external connective tissue of the muscle. So dystrophin kind of anchors this the actin filament to this complex to anchor it into the membrane and keep it stabilized. In cardiac and skeletal muscles, myosin and actin are organized in a precise array that accounts for striations. Striations are alternating A bands, dark, and I bands, light. A band is a dark band. The darkest part is where the thick filaments overlap a hexagonal array of thin filaments. H band, not as dark, it's the middle of the A band, thick filaments only. The M line is the dark transverse protein in the middle of the H band. The I band is the light band, the Z disc or Z line, protein complex that provides anchorage for thin filaments and elastic filaments. So here you can see an actual picture of individual muscle fiber or myofibrils. And here you can see one sarcomere is one contractile unit of the muscle. And here is the diagram of the contractile unit. You have a Z disc, and the Z disc is where these are anchored together. Then you have another Z disc. So inside of the Z discs is the sarcomere. Then you have the darker A band, and the A band is where you have the overlap of the myosin and actin filaments. The M line is this line in the center, to which these actin filaments are going to be pulled toward the center. These are going to be pulled toward the center to shorten the muscle and allow for the muscle contraction to happen. You have the thick filament with the myosin heads, the thin filament with actin, troponin, tropomyosin. And then here's the I band, which is lighter because it only has the actin filaments. So the sarcomere segment from Z disc to Z disc. This is the functional contractile unit of muscle. Muscle cells shorten because their individual sarcomeres shorten. Z discs or Z lines are pulled closer together as thick and thin filaments slide past each other. Neither thick nor thin filaments change the length during shortening. Only amount of overlap changes. During shortening, dystrophin and linking proteins also pull on extracellular proteins. Transfers pull to the extracellular tissue. Skeletal muscle cannot contract unless stimulated by a nerve. If nerve connections are severed or poisoned, a muscle is paralyzed. Denervation, atrophy, or shrinking of paralyzed muscle when the nerve remains disconnected. Skeletal muscle cannot contract, again, unless stimulated by a nerve, and this is repeated from the previous slide. Skeletal muscle cells are served by somatic motor neurons, nerve cells whose cell bodies in the brainstem and spinal cord that serve skeletal muscles. Their axons, called somatic motor fibers, lead to the skeletal muscle. Each nerve fiber branches out to a number of muscle fibers. Each muscle fiber is supplied by only one motor neuron. A motor unit, one nerve fiber and all the muscle fibers innervated by it. They behave as a functional unit. Muscle fibers of one motor unit are dispersed throughout the muscle, contract in unison, produce weak contraction over a wide area, and provide ability to sustain long-term contraction as motor units take turns contracting. Effective contraction usually requires contraction of several motor units at once. Average motor unit contains 200 muscle fibers distributed across as many as 100 fascicles. Motor units can vary in size. Small units provide a fine degree of control. For example, eye and hand muscles controlled by motor units with only 3 to 5 muscle fibers in each. Large motor units provide more strength than control. Powerful contractions supplied by large motor units with hundreds of fibers. For example, the quadriceps femoris and gastrocnemius controlled by motor units that may have 1,000 muscle fibers in them. So here you can see the motor unit consists of the nerve that supplies the muscle and all of the muscle fibers that are involved in that contraction. The synapse. Synapse is the point where a nerve fiber meets its target cell. When the target cell is a muscle fiber, the synapse is also called a neuromuscular junction, or motor end plate. Each terminal branch of the nerve fiber within the NMJ forms a separate synapse with the muscle fiber consisting of an axon terminal, the swollen end of a nerve fiber, which contains synaptic vesicles with neurotransmitter called acetylcholine. The nerve impulse causes synaptic vesicles to undergo exocytosis, releasing acetylcholine into the synaptic cleft. The synaptic cleft is the narrow gap between the axon terminal and sarcolemma. This slide is basically a review of what happens at the end of a nerve impulse. So once the nerve impulse reaches the axon terminals, we are going to have voltage-gated calcium channels open, calcium enters, and then that causes the release of the neurotransmitters. Then the neurotransmitters are going to go across that narrow gap between the axon terminal and the sarcolemma. So instead of stimulating another neuron like we talked about during the nervous system lecture, now we're stimulating a muscle. And the thing on the muscle is called the sarcolemma. That's the plasma membrane or the cell membrane of a muscle fiber. So when neurotransmitters are released from the axon terminals, they go across that synapse and they bind to special receptors on the sarcolemma or the cell membrane of the muscle cell. Here's a diagram of that. Here you can see motor nerve fiber. You can see all the axon terminals. If we blow up one of these and make it bigger, we can see that calcium is going to enter. Calcium causes the release of acetylcholine from the vesicles inside of the axon terminals. And once acetylcholine is released, it's going to go across the synapse and bind to specialized receptors that are in the sarcolemma. Remember, the sarcolemma is just the membrane of the muscle fiber or the muscle cell. Muscle cell sarcolemma has millions of acetylcholine receptors, proteins that respond to acetylcholine. Postsynaptic membrane folds or junctional folds of the sarcolemma increase the surface area to maximize the number of acetylcholine receptors. So here you can see it's not just a flat surface, there's actually infolding so that we can have more acetylcholine receptors here. When this axon stimulates a nerve impulse and a muscle contraction follows, we want to ensure that this muscle contraction happens. So when we release our acetylcholine here, we want lots of receptors here so that we can ensure that this muscle contraction happens. Lack of receptors causes weakness in the disease myasthenia gravis. The entire neuromuscular junction enclosed in a basal lamina, thin layer of collagen and glycoprotein separating muscle fiber and nerve ending from the surrounding connective tissue, passes through the synaptic cleft and nearly fills it. Sarcolemma and lamina with synaptic cleft contain acetylcholinesterase, an enzyme that breaks down acetylcholine, allowing for relaxation. So we're kind of just setting the stage to give you an idea of where the contraction is going to happen. Just as like a overview of where we're headed here, if we go back here to this picture, pretty far back here, there we go. So what happens is we have a nerve axon. The axon is going to lead down to an axon terminal, and the axon terminal is going to form a connection between or with the sarcolemma or the cell membrane. It's going to stimulate a local potential in this membrane, which is going to cause an electrical signal to travel through inside of the muscle fiber or the muscle cell. Once that electrical signal travels through, calcium is going to be released from these terminal cisterns. And when calcium is released, it floods the muscle. Now, we have tropomyosin blocking the myosin binding sites. When calcium is present, calcium is going to bind to troponin, which causes the shape of this to change. And when the shape changes, it's going to reveal the myosin binding sites on actin. Then what's going to happen is the myosin head can finally bind to actin, and it's going to start pulling the muscle inward. Now it's going to bind, but in order to actually cause the contraction, we need a continuous supply of ATP. So energy is going to fuel the pulling of this actin inward, and the actin on this side inward in that direction. In order to stop this from happening, or to relax the muscle, all of the calcium has to be sucked back up into the sarcoplasmic reticulum. And we have to destroy any acetylcholine that stimulated the muscle contraction in the first place. So that's where we're talking about like acetylcholinesterase. It's the enzyme that breaks down acetylcholine, allowing for the relaxation of the muscle. So the electrophysiology is the study of the electrical activity of cells. Electrical activity depends on concentration differences in ions in the intracellular fluid versus the extracellular fluid. Intracellular fluid contains greater concentration of negative anions, or negatively charged proteins, nucleic acids, and phosphates, than the extracellular fluid, and those are trapped within the cell. This creates a polarized membrane, or a net negative charge. Also, intracellular fluid has more potassium, extracellular fluid has more sodium. This is not new, so this is the exact same setting the stage as we did for the nerve impulse. Any difference in charge between 2 points is called the electrical potential. Again, review from the nerve impulse. In an unstimulated resting cell, the plasma membrane is electrically polarized or charged with a negative resting membrane potential. In a stimulated cell, ion channels in the membrane open, allowing sodium to flow into the cell down its concentration gradient, which causes depolarization of the membrane. Then, additional ion channels allow potassium to flow out, which causes repolarization. The up and down voltage shift is called an action potential. Toxins can interfere with synaptic function and can lead to paralysis. Some pesticides contain cholinesterase inhibitors, which bind to acetylcholinesterase and prevent it from degrading acetylcholine. This causes spastic paralysis, a state of continual contraction of muscles, possibly leading to suffocation. If you continually contract your respiratory muscles, you can't relax them, you can suffocate from that. Tetanus or lockjaw is a form of spastic paralysis caused by the toxin Clostridium tetani, or caused by the toxin that's produced by the bacterium called Clostridium tetani. Glycine in the spinal cord normally stops motor neurons from producing unwanted muscle contractions, but the tetanus toxin blocks glycine released in the spinal cord and causes overstimulation and spastic paralysis, or spastic paralysis of the muscles. Flaccid paralysis, a state in which the muscles are limp and cannot contract. Curare competes with acetylcholine for receptor sites, but it does not stimulate the muscles. Plant poison used by South American natives to poison blowgun darts. Botulism, type of food poisoning caused by a neuromuscular toxin secreted by the bacterium Clostridium botulinum, blocks the release of acetylcholine, causing flaccid paralysis. Botox, cosmetic injections used for wrinkle removal. So now that we've kind of set the stage and talked a little bit about some toxins that can interfere at this neuromuscular junction, now we have to talk about what happens in the muscle side of after the neuromuscular junction. The muscle contracts and relaxes in 4 stages: excitation, contraction, coupling, contraction, and relaxation. During excitation, action potentials in the motor nerve fiber lead to action potentials in the muscle fiber. So those are the events up until the action potential is sent down the T-tubule. Basically, electrical signal goes from the axon terminals, jumps across the synapse, and now it's messing with the cell membrane of the muscle cell. Excitation, contraction, coupling. So the events that link the action potentials on the sarcolemma to activation of the myofilaments, thereby preparing them to contract. In other words, converting the action potential from the T-tubule to a muscle contraction. Contraction, the step in which the muscle fiber develops tension and may shorten, also known as the cross-bridge cycle, and this leads to shortening of the muscle. And then relaxation is when the stimulation ends, a muscle fiber relaxes and returns to its resting length. So here's that nerve impulse reaching the end. We've got calcium enters due to the voltage-gated calcium channels, and then we're going to have the release of acetylcholine. Now, when acetylcholine is released, it's going to float across the synapse. This is the axon terminal. This is the muscle cell. It's going to bind to these acetylcholine receptors, and the acetylcholine receptors will open channels that allow sodium in and potassium out. This creates an end plate potential. The opening of voltage-gated regulated ion channels causes the creation of an action potential. So now we have an action or end plate potential around the membrane, the sarcolemma of the muscle cell. This action potential is going to travel down the T-tubule. And when it travels down the T-tubule, that's going to stimulate the opening of the voltage-gated calcium channels, and calcium is going to be released. Now, typically, calcium is stored in this sarcoplasmic reticulum, the terminal cistern of the sarcoplasmic reticulum to be specific. So this whole thing is the sarcoplasmic reticulum, but we've got a lot of calcium stored in this terminal cistern, and the terminal cisterns are on each side of the T-tubule. The significance of this T-tubule is it travels all the way through the muscle cell. So once we have that action potential going down the T-tubule, that's going to stimulate the release of calcium from the sarcoplasmic reticulum. And that is excitation-contraction coupling. Once calcium enters in by the filaments, calcium is going to bind to troponin. That's going to cause a shift in the shape of troponin, and that moves tropomyosin out of the way. Now, when tropomyosin moves out of the way, it exposes these myosin binding sites on actin. So now myosin can finally bind. When myosin binds, it's eventually going to be moving in a direction to pull the actin filaments toward that center, that M line, and that's going to shorten the muscle. That movement requires the hydrolysis of ATP. So first, we form that myosin-actin cross-bridge, and it produces ADP and a free phosphate. And then those, when they are released, this power stroke happens. So those come off, and the power stroke happens in this direction. Binding of new ATP breaks the cross bridge. So new ATP has to come in, and that causes this to be released. If you've ever heard of rigor mortis is the stiffening of muscles after death. So when all of your ATP is used up and you're dead, so you're not creating any more ATP, or your cells are still trying to live, but the amount of ATP that you're producing is very minimal because you don't have oxygen coming into the body anymore. So when that process happens, a few hours later, your muscles are going to contract, and they can't relax because ATP is required to detach the myosin head from actin. So the sarcolemma, or sarcoplasmic reticulum, is going to get leaky. The calcium is going to be sucked or released, and we don't have ATP present to pump that back in against its concentration gradient, so we don't have the active transport process happening to get calcium back where it needs to be. So calcium stays in this area, which constantly binds to this troponin, moving the tropomyosin out of the way, and then eventually we have the binding, and we can't release it because ATP is not present. So this is that contraction cycle, the cross bridge cycle. So really, we start by the ATP binding. When ATP binds, it kind of allows this to detach, and when it binds and it detaches, that allows for the next cycle. So then we have to cock the myosin head. Hydrolysis of this will allow the myosin head to be cocked. So we break apart ATP to make ADP and a phosphate, and then it's going to attach. Once it attaches, it has already hydrolyzed the ATP into ADP and PI, and then you're going to have the power stroke happen. When the power stroke happens, these guys go away, and the myosin head does its power stroke, which moves the actin in a particular direction. And then in order to detach this and reset it, we have to have more ATP come in and do that. During relaxation, we have to relax this on two fronts. First, we have to stop stimulating the cell membrane of the muscle cell. Second, we have to stop the calcium from binding to troponin. So, here we go. We have acetylcholine. We stop releasing acetylcholine into the neuromuscular junction. And then we have this enzyme, acetylcholinesterase, that's going to destroy any acetylcholine that was released, or that still remains in that synapse. Then we're going to have the calcium actively reabsorbed by the sarcoplasmic reticulum. When calcium leaves, then tropomyosin blocks those actin binding sites, or the myosin binding sites on actin. This returns tropomyosin to the position blocking active sites of actin. Length tension relationship. The amount of tension generated by a muscle depends on how stretched or shortened it was before it was stimulated. If overly shortened before stimulated, contraction is weak. Thick filaments butt up against Z discs. If too stretched before it's stimulated, the contraction is weak. Minimal overlap between thick and thin filaments results in minimal cross-bridge formation. The optimum resting length produces the greatest force when the muscle contracts. Nervous system maintains muscle tone or partial contraction to ensure that resting membranes are near this length. So here you can see overly stretched, they're not really overlap that much, so that's a problem. Overly contracted, they're too contracted, you can't really contract it anymore. This is your optimum resting length. Rigor mortis, I already talked about a little bit. So that's the hardening of muscles and stiffening of the body beginning 3 to 4 hours after death. Deteriorating sarcoplasmic reticulum releases calcium, and deteriorating sarcolemma allows calcium to enter the cytosol. Calcium activates myosin, actin cross-bridge formation, and muscle contraction. Muscle relaxation requires ATP, and ATP production is no longer produced after death. So the fibers remain contracted until the myofilaments begin to decay. Rigor mortis peaks at about 12 hours after death and then diminishes over the next 48 to 60 hours. A muscle twitch. So a quick cycle of contraction and relaxation when the muscle is directly stimulated with an electrode. On a myogram, it's a graph that measures the twitch. It can show 3 phases. The latent period, which is the delay just after stimulation of the muscle, the time required for excitation-contraction coupling, and tensing elastic components, generates internal tension, which is not visible in the myogram. Contraction phase, the external tension is generated and load is moved as the muscle fiber shortens. The relaxation phase, sarcoplasmic calcium levels fall as calcium is reabsorbed into sarcoplasmic reticulum. The tension declines. So here's the latent period, the contraction phase, and relaxation phase that you can see on a myogram. Threshold is the minimum voltage that causes a muscle twitch. Even if the same voltage is delivered, different stimuli cause twitches in varying strengths because the muscle's starting length influences tension generation. Muscles fatigue after continual use. Warmer muscles enzymes work more quickly. Muscle cells' hydration level influences cross-bridge formation. Increasing the frequency of stimulus delivering increases tension output. Muscles must contract with variable strength for different tasks. Stimulating the nerve with higher voltages produces stronger contractions. Higher voltages excite more nerve fibers, which stimulate more motor units to contract. Recruitment, or multiple motor unit summation, the process of bringing more motor units into play with stronger stimuli, occurs according to the size principle. Weak stimuli, low voltage, recruit small units, while strong stimuli recruit small and large units for powerful movements. And here you can see where thresholded is and the proportion of nerve fibers excited and the response of the muscle. Stimulus and intensity contraction strength. So the frequency of stimulation also affects twitch strength. Low frequency stimuli produced in identical twitches, identical twitches. Higher frequency stimuli, for example, 20 stimuli per second, produce temporal wave summation. Each new twitch rides piggyback on the previous one, generating higher tension. Only partial relaxation between stimuli resulting in fluttering, incomplete tetanus. Unnaturally high stimulus frequencies in lab experiments cause a steady contraction called a complete or fused tetanus. So here you can see individual muscle twitches. Here is temporal summation, where you have an increase and the other one kind of piggybacks on it, so it just keeps going up and up and up. And complete fused tetanus. So if you continuously stimulate it, it will just stay contracted. Isometric versus isotonic contraction. Isometric contraction is a contraction without change in length. Muscle produces internal tension, but external resistance causes it to stay the same length. Can be a prelude to movement when tension is absorbed by elastic component of the muscle. It's important in postural muscle function and antagonistic muscle joint stabilization. Isotonic contraction, contraction when a change in length but no change in tension. Muscle changes the length with no change in the tension. There's concentric contraction where muscle shortens as it maintains tension, for example, lifting weights. And then there's eccentric contraction where the muscle lengthens as it maintains the tension, for example, slowly lowering the weight. And this is repeated here. So here is a picture of isometric contraction. Isometric contraction, you are not moving the weight. So the muscle develops tension but does not shorten. Isotonic concentric contraction, that's when you have shortening of the muscle. Isotonic eccentric contraction, that's when you have lengthening of the muscle. The tension is the same because you have the same weight in these situations, but one of them causes tension that shortens the muscle, the other one causes tension that lengthens the muscle. And the length of tension versus time is depicted here. Some people like looking at graphs, some people the graphs are more confusing, so if this is helpful to you to understand it, by all means use it. If it's not helpful, you can kind of skip past this one. Muscle metabolism, so ATP sources. All muscle contraction depends on ATP. ATP supply depends on the availability of oxygen and organic energy sources. For example, glucose and fatty acids. Two main pathways of ATP synthesis would be anaerobic fermentation, that enables the cells to produce ATP in the absence of oxygen. It yields little ATP and lactate, which needs to be disposed of by the liver. And aerobic respiration, which produces far more ATP and does not generate lactate, requires a continual supply of oxygen. So aerobic respiration, using oxygen from myoglobin, that's the oxygen that's stored in the muscles, that happens immediately and diminishes pretty quickly. Then you have the phosphagen system, and then the glycogen lactate system, anaerobic fermentation. And then eventually you need to switch to aerobic respiration, which is supported by cardiopulmonary function. So immediate energy for short, intense exercise, oxygen is briefly supplied by myoglobin, but it's rapidly depleted. Myoglobin is that protein that stores oxygen in your muscles. Muscles meet most ATP demand by borrowing phosphate groups from other molecules and transferring them to ADP. There's two systems that control these phosphate transfers, the myokinase and creatine kinase. Myokinase transfers phosphate from one ADP to another, converting the latter to ATP. Creatine kinase obtains phosphate from a phosphate storage molecule called creatine phosphate, and it gives it to ATP. So this is why some bodybuilders will consume creatine, because creatine can't store phosphate groups, which allows you to maintain your muscle contraction for a longer period of time, or decreases the time to fatigue for a muscle. In the phosphagen system, the combination of ATP and CP, which provides nearly all energy for short bursts of activity. These are the phosphagen systems, so you can see the myokinase, creatine kinase. As the phosphagen system is exhausted, muscles shift to anaerobic fermentation. Muscles obtain glucose from blood and their own stored glycogen. In the absence of oxygen, glycolysis can generate a net gain of 2 ATP for every glucose molecule consumed. Converts glucose to lactate. Anaerobic threshold or lactate threshold, the point at which lactate becomes detectable in the blood. Glycogen lactate system, the pathway from glycogen to lactate, produces enough ATP for 30 to 40 seconds of maximum activity. For long-term activity, after about 40 seconds, the respiratory and cardiovascular systems start to deliver oxygen fast enough for aerobic respiration to meet most of the muscle's ATP demand. Aerobic respiration produces more ATP per glucose than glycolysis does, efficient means of meeting the ATP demands of prolonged exercise. After 3 to 4 minutes, the rate of oxygen consumption levels off to a steady state where aerobic ATP production keeps pace with demand. For 30 minutes, energy comes equally from glucose and fatty acids. Beyond 30 minutes, depletion of glucose causes fatty acids to become the more significant fuel. Muscle fatigue, progressive weakness from prolonged use of muscles. Fatigue and high-intensity exercise is thought to result from potassium accumulation in the T tubules, which reduces excitability, ADP and phosphate accumulation, which slows cross-bridge movements, inhibits calcium release, and decreased force production in myofibrils. Fatigue and low-intensity, long-duration exercise is thought to result from fuel depletion, electrolyte loss, and central fatigue. Maximum oxygen uptake, or VO2 max, the point at which the rate of oxygen consumption plateaus and does not increase further with added workload. Major determinant of one's ability to maintain high-intensity exercise for more than 4 to 5 minutes. If you are interested in exercise physiology, this is going to be one of the fundamental concepts that you will learn through your studies with that. People dedicate entire careers to altering someone's VO2 max because their VO2 max determines their level of fitness and their level of ability to do prolonged exercise. This is proportional to body size and peaks at around age 20, usually greater in males than females, can be twice as great in trained endurance athletes than in an untrained person. Excess post-exercise oxygen consumption, or EPOC, oxygen debt, is the elevated rate of oxygen consumption following exercise. The purpose, aerobically replenish ATP, replace oxygen reserves on myoglobin, provide oxygen to the liver that's busy disposing of lactate, and provide oxygen to many cells that have elevated metabolic rates after exercise. EPOC can be 6 times basal consumption and last an hour. So physiological classes of muscle fibers. There's slow twitch, fast twitch, different types of muscle fibers that we have. So we're going to go through what they are and what they're used for. So the first one, slow twitch, slow oxidative, or SO, red fibers, or type 1 fibers. They're well adapted for endurance. They resist fatigue by oxidative ATP production. They're important for muscles that maintain posture. Thin cells with abundant mitochondria, capillaries, myoglobin, and contain a form of myosin with slow ATPase and an SR that releases calcium slowly. Grouped in small motor units controlled by small, easily excited motor units allowing for precise movements. Fast twitch or fast glycolytic white fibers. Fibers well adapted for quick responses utilize glycolysis and anaerobic fermentation for energy. They're abundant in quick and powerful muscles, eye and hand muscles, gastrocnemius of the calf, and biceps brachii. They contain a form of myosin with fast ATPase and a large sarcoplasmic reticulum that releases calcium quickly. Fibers are thick and strong. Low myoglobin gives them a pale color. They're grouped in large motor units controlled by larger, less excitable neurons, allowing for powerful movements. There's an intermediate or fast oxidative fiber. That type is known mainly in other mammals, but relatively rare in humans. So here are the different types of muscle fiber types. So within one fascicle, you would have these different types. Every muscle contains a mix of fiber types, but one type predominates depending on the muscle function. The fiber type within a muscle differs across individuals. Some individuals seem genetically predisposed to be sprinters, while others more suited for endurance. Factors that affect muscle strength. Muscle size. The thicker muscle forms more cross bridges. A muscle can exert a tension of 3 or 4 kilograms per centimeter squared of cross-sectional area. Fascicle arrangement. Pennant are stronger than parallel, and parallel stronger than circular. Size of active motor units. The larger the motor unit, the stronger the contraction. Multiple motor unit summation. Simultaneous activation of motor units increases the tension. And temporal summation. The greater the frequency of simulation, the stronger the muscle contraction. The length-tension relationship. A muscle resting at optimal length is prepared to contract more forcefully than muscles that extensively contracted or stretched. That's something we talked about previously. Fatigue. Fatigued muscles contract more weakly than rested muscles. Resistance exercise. Contraction of a muscle against a load that resists movement. Example, weightlifting. Growth is from cellular arrangement. Muscle fibers synthesize more myofilaments and myofibrils and grow thicker. Endurance aerobic exercise, contraction of a muscle against a load that resists movement. Example, weightlifting. Improves fatigue resistance of muscles. Increases skeletal strength, red blood cell count, and oxygen carrying capacity of the blood. Enhances cardiovascular, respiratory, and nervous system function. Cardiac and smooth muscle share certain properties. Their cells are myocytes, not as long and fibrous as skeletal muscles. They're relatively short and have 1 or 2 nuclei. Cardiac muscle cells are specifically called cardiomyocytes. They are involuntary, not usually under conscious control. They receive innervation from the autonomic nervous system. Cardiac muscle contracts with regular rhythm, works in sleep or wakefulness without fail and without conscious attention. Highly resistant to fatigue, muscle cells of a given chamber must contract in unison. Contractions must last long enough to expel blood. They're striated, relatively short and thick, surrounded by endomysium but no paramecium or epimecium. Cardiomyocytes branch and join together. Others are joined to other cells at connections called intercalated discs. They appear as thick, dark lines on stained tissue sections. That's where the electrical gap junctions allow for each cardiomyocyte to directly stimulate its neighbor. Mechanical junctions that keep the cardiomyocytes from pulling apart. No satellite cells and very limited mitosis, so cardiac muscle cells repair by fibrosis. After a heart attack, functional muscle is not regenerated. Fibrous connective tissue is created in its place. They can contract without the need for nervous stimulation, contains a built-in pacemaker that rhythmically sets off a wave of electrical excitation. Wave travels through the muscles and triggers contraction of the heart chambers. Heart is autorhythmic, able to contract rhythmically and independently. Stimulation from autonomic nervous system can modify the contraction rate and strength. And they're highly resistant to fatigue. Smooth muscle. So it's named for its lack of striations. Innervation varies. Some smooth muscles lack nerve supply, others receive input from autonomic fibers, with many varicosities, containing synaptic vesicles. Capable of mitosis and hyperplasia, injured smooth muscle regenerates well. Contraction is slower than skeletal and cardiac muscle, takes longer to contract, but can remain contracted for a long time without fatigue. So this shows a diagram of the innervation of and varicosities of smooth muscle. Forms layers within the walls of hollow organs. It can propel contents of an organ, for example, food in the digestive tract. It can modify pressure and flow of blood in the circulatory system. So surrounding the walls of the blood vessels. Can provide fine control in some locations. Smooth muscle contractions of the iris controls pupil size. This is a diagram showing the cross-section of the lower part of the esophagus. And you can see the muscle layers diagrammed here. So here's the smooth muscle layer. Myocyte structure, so for smooth muscle, they're fusiform shaped. In other words, they're wide at the middle and they have tapered ends. They have a single nucleus located near the middle of the cell. Thick and thin filaments are present, but they're not aligned with each other. There's minimal sarcoplasmic reticulum and no T-tubules. Calcium is needed for muscle contraction, comes from the extracellular fluid by way of channels in the sarcolemma. Z-discs are absent and replaced by dense bodies, protein masses that form plaques on the interface of the plasma membrane and scattered throughout the sarcoplasm. Dense bodies of neighboring cells are linked together. There's multi-unit and unitary or single-unit smooth muscle. Multi-unit smooth muscle, individual myocytes respond independently of all the others. Autonomic innervation forms motor units, terminal branches of a nerve fiber, synapse with individual myocytes. Occurs in some of the largest arteries and air passages. Unitary or single unit smooth muscle, myocytes are coupled by gap junctions, so they contract as a unit. Most common type present in the digestive, respiratory, urinary, reproductive tracts, so also called visceral muscle. And this shows a picture of multi and unitary smooth muscles. Excitation of smooth muscle. Smooth muscle can contract or relax in response to a variety of stimuli. Autonomic nerve fibers and neurotransmitters, for example, parasympathetic nerves secrete acetylcholine, stimulating GI tract smooth muscle, and sympathetic nerves secrete norepinephrine, relaxing smooth muscle in bronchioles. Chemicals, smooth muscle responds to hormones, oxygen, low pH, and others. Temperature, for example, cold excites piloerector muscles, and warmth relaxes muscle and skin and blood vessels. Stretch, for example, the stomach contracts when stretched by food. Autorhythmicity, for example, some single unit smooth muscle cells in digestive tract depolarize at regular intervals. Contraction is always triggered by calcium, energized by ATP, and achievement by sliding filaments. Smooth muscle gets most calcium, as I mentioned, from extracellular fluid. Calcium channels are concentrated in calveolae, pockets on the sarcolemma. Their different channels, gated by different stimuli, can respond to mechanical stretch, voltage, or chemical ligands. No troponin. Calcium instead binds to calmodulin. Calmodulin then activates myosin light chain kinase, which adds a phosphate to the regulatory protein on the myosin head. Thick filament pulls on the thin ones pull on dense bodies and the membrane plaques. It twists kind of like wringing out a wet towel. So here you can see how they're all pulling against each other and what contracted smooth muscle would look like. Contraction and relaxation is very slow in comparison to skeletal muscle. There's a latent period in smooth muscle. Tension peaks at about 500 milliseconds. Due to slow myosin, ATPase enzyme, slow calcium pumps, and calcium binds to calmodulin instead of troponin. The latch bridge mechanism is resistant to fatigue. Heads of myosin molecules do not detach from actin immediately. They do not consume any more ATP. Maintains continual tonic contraction or smooth muscle tone. Stretch can open mechanically gated calcium channels in the sarcolemma, causing contraction. Stress relaxation response helps hollow organs gradually fill, for example, the urinary bladder. Unlike skeletal muscle, smooth muscle contracts forcefully even when greatly stretched. It exhibits plasticity, the ability to adjust its tension to the degree of stretch. A hollow organ such as the bladder can be greatly stretched, yet not become flabby when empty. Able to contract forcibly or forcefully when stretched because there are no Z-discs, so thick filaments cannot butt against them and stop contraction. Since the thick and thin filaments are not arranged in orderly sarcomeres, stretching does not cause a situation where there's too little or too much overlap. The thick filaments of smooth muscle have myosin heads along their entire length, so the cross bridges can form anywhere. And then here's a really good table that kind of explains the differences between skeletal, cardiac, and smooth muscle contractions, so please take some time to review this table. We're just going to talk about two diseases briefly that affect the muscular system. Muscular dystrophy, it's a group of hereditary diseases in which skeletal muscles degenerate and weaken and are replaced with fat and fibrous scar tissue. Duchenne muscular dystrophy is caused by sex-linked recessive trait. Most common form, diagnosed around 5 years of age, mutation in gene for muscle protein dystrophin, actin, not like sarcolemma, and cell membranes are damaged during the contraction. Necrosis and scar tissue result. Rarely live past 30 years of age due to the effects on respiratory and cardiac muscle, and it is incurable. Myasthenia gravis is an autoimmune disease in which antibodies attack the neuromuscular junctions and bind acetylcholine receptors together in clusters. Muscle fibers then remove the clusters from the receptors from the sarcolemma by endocytosis. The fiber becomes less and less sensitive to acetylcholine. Effects usually first appear in facial muscles, drooping eyelids, double vision, difficulty swallowing, and weakness of the limbs. Usually occurs in women between 20 and 40 years of age. Treated with drugs to reduce acetylcholine breakdown or suppress the immune response. And this shows at the start, at 60 seconds, and at 90 seconds.